All right, back again. We're going to talk about how a protein is made now. Where we left off from the mRNA or the RNA notes, we talked about how the DNA gets copied by RNA polymerase, and then the mRNA breaks away from the DNA, heads to a ribosome in the cytoplasm. Before we go any further, we just have to mention a couple things. What is the genetic code specifically? It's combinations of 3 RNA nucleotides that are coding for specific amino acids. When we put amino acids together, we build a protein. And that's all the DNA does, is tells us how to build proteins. Proteins, for example, in my eyes make my eyes green. Proteins decide whether or not we have cancer or how tall we get or what color our skin is. So that's all DNA is telling us, is how to build proteins. And the way we know how to build proteins is this pattern of letters. It's not a pattern, this combination of letters in the RNA that tells us which amino acids to pull up. 3 nucleotides put together are called codons. It's like it's a secret code, and you get those codons on an mRNA strand. Let me show you the table for mRNA. There are 64 possible combinations of the 4 letters A, C, G, and U in 3-letter groupings. So there's 64 possibilities. And so you can see on this table in the different combinations which ones call for which amino acids. So the cell knows specifically when it reads, for example, in the upper left-hand corner, when the ribosome reads UUU, it knows that it needs to bring over a tRNA that is carrying the phenylalanine amino acid. So this is the table. You can see that there is 1 start codon. It's at the beginning of every gene. At the beginning of every mRNA, it's AUG. And then there are 3 different stop codons.
Good morning, staff. If you have seniors in your Monday advisory, please pick up the handouts in the main office. Thanks.
Okay, that was fun to have an announcement in the middle of my notes. Okay, protein synthesis. This is where the proteins are actually made. Translation is the process of making proteins from an mRNA strand. It sounds like the name transcription. So look at the diagram. Transcription is making the RNA. Translation is actually building the protein at the ribosome. So after leaving the nucleus, the mRNA travels to a ribosome. And there are many different ways to draw ribosomes. They have 2 parts to them, a small subunit and a large subunit, and they're awkward to draw. So here's a couple different images of ribosomes, the mRNA traveling through, the trucks pulling up with their amino acids. For the purposes of learning, I'm going to simplify that drawing incredibly. The ribosome is these 2 rectangles stacked on top of one another. And then you see the mRNA traveling across the bottom, the A's, U's, G's, and C's all together. So it says the mRNA moves through the ribosome 3 nitrogen bases at a time. And we just learned that those 3 bases are called a codon. And notice that I correctly designed this, that the first 3 letters of mRNA are the start codon. When the ribosome reads the AUG start codon, it calls over the first truck that has the opposite or complementary 3 letters. So notice that the truck, it's anti-codon. Anti means the opposite of the codon. Has a U, an A, and a C, the opposite 3 letters of the AUG codon. So it pulls it over and it carries with it the first amino acid. The ribosome then moves to the next codon and reads the codon and calls over the next truck with the correct anti-codon, and it's carrying a specific amino acid. The ribosome continues to move down 3 letters at a time, calling over the trucks 1 at a time, carrying their amino acids. This continues until all the amino acids are in place. The tRNAs fall away, or I say the trucks drive away, leaving their building supplies behind, and the amino acids bond together. What we end up with at the end is a finished product called a protein. The protein detaches and the mRNA molecule leaves. The mRNA molecule can be read over and over and over again. The trucks can be used over and over and over again. This is a great video that I really recommend that you watch, but we're not going to watch it right here. Here's a super diagram that came from our textbook that looks at translation from beginning to end. So you see the big red blob that looks like the moon. You see an mRNA coming out of that. So it's been copied during transcription, copied one side of the DNA. It's leaving and it's heading to this blob, which is the ribosome. And then you can see the trucks, and some of them are carrying amino acids. They come over when they have the correct opposite 3 letters, and they drop off their amino acids. And then that chain of spheres put together is the building protein. I often like to use a little analogy to try to help people remember the different steps of protein synthesis or translation. So here's my little analogy. Remember that the DNA has to stay tucked safely in the nucleus. I like to think of DNA as the architectural designs for an office building. They're stored on a computer and they are super important, and you don't want to lose those original designs because then the architect would have to start all over and the building would be conceivably ruined. The nucleus is the architect's office, and it's a safe place to store the designs. The mRNA, remember mRNA is a copy of one side of the DNA. The mRNA is like a bike messenger carrying blueprints or copies of the original plans to the construction site. It's possible that something bad could happen to that bike messenger and his or her blueprints. It could rain and wreck the blueprints, or the bike messenger could, get hit by a car. If that happened, the original architectural designs could be printed again, and another message could be sent out of the nucleus and to the ribosome. Okay, the ribosome is the construction itself, the construction site itself. I called it the construction worker here, who's reading the blueprints and determines what building materials are added when and where. When you're building a new office building, you have to build the structure of the building before putting in the carpeting, for example. So we've got to make sure that the amino acids are coming in the correct order. They can't come in the wrong order. The tRNA, again, are the trucks that are carrying the building supplies. Each truck is labeled with an anti-codon, so we know what it's carrying, and the trucks can be used over and over again. They drop off their supplies, they go back and pick up the same supplies. So a plumbing truck is never going to go pick up carpeting. It's always going to pick up plumbing supplies. And then the amino acids represent the building materials for the building. So concrete, pipes, carpeting, those are the amino acids. They have to be added to the building in the correct order, or the building would be a disaster. And finally, the protein would be the finished office building, where all the supplies were put together in the correct order, and you have a protein. That's it.